The overall goal of the following experiment is to utilize high-content assays in cellular disease models to quantify pathways affected in neurodegenerative diseases. To accomplish this, a cellular model of the disease phenotype is input into the automated cell culture system, and cells are incubated until the required cell number is reached. Cells are then split, counted, and dispensed into 96-well plates containing lentivirus expressing various shRNAs. After 6 days of cellular differentiation, cells are output from the system for immunostaining and high-content imaging. Finally, the images are analyzed using a variety of analysis algorithms to quantify features related to cell health, neuronal outgrowth, and mitochondrial function. Results are also analyzed by two-way ANOVAs to identify significant interaction effects.
This method can expedite answering key questions as to the function of disease-related genes and the pathways within which they function. Overall, this will then lead to a better understanding of the pathogenic mechanisms involved in neurodegenerative diseases.
To begin the cell culture process, prepare the automated cell culture system for the input of cell culture plates. Load consumables such as pipette tips, cell culture plates, and assay plates into the system. Also, ensure there is sufficient cell culture media, PBS, and trypsin in the system. Manually seed 2 omni-tray plates with 2 × 10⁶ cells per plate of the SHSY5Y neuroblastoma cell line. Maintain cells in optimum media with 10% fetal bovine serum, FBS. Place the